Hello, and welcome to the second video in the second and final video in this series on DNA structure and replication. And I'll try to keep this as short as I can because I know that other video was already like a 15 and change minutes. So I'll try to keep this as brief as I can. So in this video, we're going to be talking about DNA replication, and it's somewhat important that you've watched, actually it's really important that you've watched the previous video already, because as we're going through DNA replication, the structure of DNA is actually going to be really important in terms of how it replicates. So we'll get into that in a second. But first, I just want to go over what DNA replication even is and make sure we all know what process we're talking about when we're talking about DNA replication. So hopefully this concept of making a copy of the DNA, which is what DNA replication is somewhat familiar to you already. As we were going and talking about mitosis, we talked about how every cell in preparation to divide needs to make a copy of its DNA so that the daughter cells get the full complement of chromosomes, the full set of genetic material. So anytime mitosis is going to occur, whether that's because of asexual reproduction, whether that's because of growth, whether that's because of something like wound healing, any situation where mitosis is going to happen, DNA replication also needs to happen. And we also should hopefully know that DNA is being replicated during the S phase of interphase. So in that period where the cell is growing and doing its duties and making proteins and all of that, in that interphase period, there's a section called S phase where the DNA is copied. So let's take a look at how exactly this happens. So remember that in order to copy our DNA, we need it's sort of very similar to making a copy of the cell, right? We're taking a parent cell and creating two daughter cells. And for DNA, we're taking parent DNA and making two, you could think of it as daughter DNA molecules. So in that way, it's very analogous to what happens with cells. And the way that this happens is that we use the parent DNA as a template to create the new DNA. And so let's look at this. So remember that for each DNA has two strands, right? And the nitrogenous bases that are connected to each nucleotide, we said that they're complementary to one another. So where we find a G, we'll also find a C. Where we find a T, we'll also find an A. And so what's going to happen is that the two strands of the parent DNA are going to separate. And this is possible because the bonds that are holding the two strands together are those weak hydrogen bonds. It's really, we said, just an attraction. So it's relatively easy to pull the two strands of the parent DNA molecule apart from one another and separate them. And once they're separated, each strand is able to act as a template for the daughter molecule. So the way that this works is if we have a C on the parent strand, then we know that we need to have a G on the other side. Here we have an A, so we know that we need to put a T right here. Here we have a C, so the daughter strand knew that it needed a G. Here's a G and it needed a C. So we're able to create a copy of what the original parent DNA molecule was by just using the complementary base pairing rules that we already talked about in the last video. And the same thing is going to happen on this strand over here, exactly the same. We have one parent DNA strand and it's being used as a template. So here the parent DNA strand had a G. The daughter strand was knew that a C needed to be across from it. We've got a T here and we'll need to put an A here. And so if you compare the two new pieces of DNA, you can see that they're identical to both each other. So GC, GC, TA, TA, GC, GC, CG, CG. So they're identical to each other, but they're also identical to what the parent cell or what the parent DNA strand had as well. So so we get two identical copies because of this template mechanism. So we start with one parent DNA molecule and we end up with two daughter DNA molecules. And in terms of their composition, each daughter DNA molecule is going to be made up of half of the original parent strand. So this blue color represents the original parent, and each daughter DNA molecule is going to have one blue strand, but also half new material that was just created in the copying process. So that's shown here in yellow. So each daughter DNA molecule is going to have half original and half new molecules. And the word for this is semi-conservative. So it's sort of the original DNA is half conserved. You know, each daughter DNA molecule has half the original material. So it's semi-conservative. Okay. So here we're going to take a look at the players that are involved with DNA replication, the enzymes. We're going to keep it pretty simple and only talk about two of them in detail. And those two enzymes are helicase, which you can see here, and DNA polymerase, which you can see here. So helicase is important for the first role because it's going to sort of unwind the DNA and open up those two strands, separate them from one another. So that's what helicase is going to do. And it's going to create this opening or this bubble of the DNA. And then once the new DNA or once the new nucleotides come in, because we are creating a new strand, so once the original parent strands are separated from one another, we need to make the other side, right? We need to get the complementary bases and make the new strand on the opposite side, right? And so an enzyme called DNA polymerase is going to be responsible for this. It's going to take the new nucleotides and attach them to one another to make the new strand. So it's not connecting the two nucleotides together, because remember, these are only connected because of hydrogen bonds. Rather, for the new strand, for the new strand that's being formed right here, it's connecting this nucleotide to this nucleotide, and then it'll add this nucleotide on and connect it to the new strand. So it's creating a new sugar phosphate backbone with the associated nitrogenous bases. So this is shown here in words instead of pictures in case that speaks to you. So the two enzymes, one is helicase, and that's the job for that one is to unzip the parent DNA strands. And then those free nucleotides are going to come in and pair with their complementary nucleotides on the parent strands. And then DNA polymerase is going to make bonds between the new nucleotides on the new strand. So it's nucleotide to nucleotide that the DNA polymerase is connecting. Okay. So here's this question of where DNA replication starts. And it's a little bit different for prokaryotes and eukaryotes. Maybe you remember that prokaryotes have a single circular chromosome. So their DNA is in a circle and it's just one piece. And so for prokaryotes, what happens is there's just a bubble that opens up because helicase separated the two strands. And then the bubble sort of grows as DNA replication occurs on both pieces. And it just keeps going further and further and further until the whole circle, the whole DNA piece is replicated. But for eukaryotes, they have a lot more genetic material. So if they were to open up just one bubble and go out from there, it would take forever and ever. It would take hours and hours for the whole piece of DNA to be replicated, just because it's so long and there's so much DNA. So what happens instead in eukaryotes is that we have multiple points where replication starts. And we call the starting point of replication the origin of replication. Origin of replication is just the starting point. And it creates these bubbles. We can call them replication bubbles. And so we're going to start here and open up a bubble, open up the two strands in both those places. And then we'll go out from those bubbles in both directions. And eventually they'll meet in the middle. And then this whole chunk of DNA will be replicated, right? So the end result is that we're going to get two new strands of DNA that are both, I guess, two new pieces of DNA that are both double stranded. And the whole point of this is to speed up the process by introducing these multiple bubbles, multiple origins. Okay, and this is the last slide. So I just want to bring this up because it will be a little bit important in next semester when we start talking about evolution. So when DNA is being copied, there's always the possibility that DNA polymerase or that the free nucleotides are going to be incorporated incorrectly, that we're going to get a wrong base, a wrong nucleotide. So for example, here we have a T, so it should be an A right here, but instead you can see that there's a G. So sometimes we get mistakes and it causes what's called a mutation. And these mutations can be really damaging and dangerous to an organism if they're not fixed. So what actually happens is that your cells, it's so important to get this process right, to copy the DNA faithfully, that the DNA has these repair mechanisms, or that the cells have these repair mechanisms to sort of do a surveillance check of the DNA and make sure that everything is being copied correctly. So it's sort of like proofreading when you're about to submit an essay or a paper, you want to make sure that it's free of mistakes. The same thing is happening with the DNA. It's checking to make sure that the bases are correct. And if it's incorrect, we have special repair enzymes that can correct the errors that it finds. They're actually special versions of DNA polymerase that can fix the problems. So we get a replacement base, and then we get now both the strands are perfectly aligned with the proper base pairing rules. So DNA repair is a process that happens in all of our cells, and it catches a lot of the mistakes that can be incorporated during the copying of DNA. Of course, some mutations will slip through, some mistakes will slip through, and that's just an inevitable thing that's going to happen every time our DNA is replicated. But a lot of the mistakes are caught with these DNA repair and proofreading systems. Okay, that concludes our final video lecture series for this semester. So I know you guys are so sad about it. I will see you soon.